Bio30, welcome to the first part on protein synthesis. This is transcription to translation, how we make proteins in the body. Please follow along, filling in any bolded words or underlined words within your notes. So we're going to start at the top with protein synthesis and what is it? So we know nitrogen-based organic compounds are made of amino acids. That's why when we learn in the digestive unit that proteins break down into amino acids. We also learn in chapter 9 with the kidneys that when proteins break down, it creates a nitrogenous waste, which is dangerous for our body. So proteins are made out of nitrogens, which break down into amino acids. We're going to talk about the process of going from amino acids to proteins. 30%, 40% of your body weight is approximately proteins. And as you can see there, I have a long list of examples of proteins that make up your bodies. Enzymes, hormones, membranes, transport proteins, structural proteins, and antibodies. You don't have to write all those examples down. Maybe pick or choose one or two just as a reminder to yourself. So we need to discuss an amino acid. Bio20, you learn that proteins break down into amino acids. It's a simple macromolecule. It's the smallest form of a protein. But what actually is an amino acid? They're made of carbon, hydrogen, oxygen, and nitrogen, and they're essentially the building blocks of all proteins. Now, the cool thing about amino acids is there's only about 20 of them. So that means that there's a lot of redundancy when we have to make proteins. So due to the only 20 different amino acids, it's easy to kind of memorize them. And in university, that'll be something you'll do in certain classes. Which leads me to, this is kind of an interesting fact about an amino acid, is that 8 are essential and cannot be produced by the human. They have to be consumed. So there's a lot of different ones that you don't produce naturally. You have to actually eat them. Something, for example, like tryptophan is an example of an amino acid. You eat tryptophan when you eat turkey. Kind of one of the reasons why it gets retired in turkey. So let's talk about DNA to proteins. You know DNA belongs in your nucleus. You know DNA is made up of A, T, C's, and G's, and you know that we can replicate it. You know that a cell as a whole can go through mitosis and meiosis. But how do you go from DNA to actually what builds you, a protein? So what we have to go through is protein synthesis. Every DNA triplet, now a triplet, remember, is 3 letters long. CTA is an example, okay? Every DNA triplet actually represents one type of amino acid. And the genetic code determines the sequence of amino acids to make that protein. So your DNA tells your body what proteins to make. Every gene on the chromosome at least makes one protein. And in order to make proteins, DNA has to make a copy of itself in something called RNA, something new that we don't know, but we're going to learn about it. RNA is single-stranded, it's not double-stranded like DNA, and it has some different characteristics. You will be getting one of these charts. I will post it in our Google Classroom. Please have it readily at all times for the rest of this chapter, as this is going to be the bulk of what we are doing from now on. So this is called an amino acids chart. And essentially what it does is it takes what we call messenger RNA, which I will discuss with you in a minute, and tells you how to figure out what amino acids belong to it. So let's say I had an triplet or a codon that is GAC, all right? How I read this chart is I'm going to always go to the first base. So I said G first, so here's my G. I'm then going to go to the second base, which is located up here, and I'm going to find my A. So it should fall in this square here. The third base, I can follow along, find my C, and there it is. GAC is aspartate, okay? So that's how you read these using messenger RNA. Well, what is messenger RNA? I'm going to get to that in a minute. In your notes, you have a comparison chart of DNA to RNA to protein. So what I want you to know is that DNA, which we've learned about, is only located in the nucleus. It never, ever, ever leaves the nucleus unless it's going through mitosis and meiosis, in which the nuclear membrane breaks down and then it's kind of floating in the cytoplasm. But otherwise, the nucleus is where DNA is housed, okay? Think of DNA like a master blueprint. If I was to give away the master blueprint to a project I'm working on and just give it away to someone and I didn't have it, I wouldn't know what I looked like or what I was building. So I need to make a copy of that. And that was what RNA does. RNA is the copier of DNA. DNA is double-stranded. We know it has complementary strands, right? A's goes with T's, C's goes with G's. It's made up of deoxyribose, so nitrogen bases, A, C's, G's, and T's. RNA, on the other hand, is a little bit more free. It can travel between the nucleus and the cytoplasm. And the nice thing is it is single-stranded. It only has one strand to it. It doesn't have a deoxyribose base, it actually just has what we call ribose. And you'll notice that in the nitrogenous bases, that T from DNA gets switched out for something called U or uracil for RNA. There are 3 types of RNA that we are going to go through as well and add to your notes. So understanding that DNA is copied by RNA to make proteins. So what are the types of RNA that we have in our body? Well, one of them is called messenger RNA. This is going to be the most important RNA for the rest of this chapter that you need to know. So messenger RNA, or mRNA as I will refer to it, is involved in the process of transcription, which is essentially the first stage of protein synthesis. Its job is to copy DNA. That is our mRNA's only job, is to create the message or the copy that needs to leave the nucleus. So the DNA is stuck in the nucleus, and we need to get from the DNA in the nucleus, we need to make that copy to get it to something called the ribosome. So mRNA can be found in both the cytoplasm and the nucleus of your cell because it travels between both. rRNA, or ribosomal RNA, is essentially what makes up the ribosome. Science 10, you learned about a ribosome. Ribosomes deal with proteins, and this is what we're going to talk about. It's going to become something very important as we move into translation. The last one we're going to talk about is transfer RNA, or tRNA, and it carries amino acids. So amino acids just don't randomly show up to the party. They need to be invited. So transfer RNA is their invitation to the party. And transfer RNA brings amino acids to the mRNA during the second stage of protein synthesis, which is called translation. Now, rRNA and tRNA are only found in the cytoplasm. They don't travel back and forth between the nucleus and the cytoplasm. So here's another way to look at DNA versus RNA. DNA is double-stranded, okay? RNA is single-stranded. When we copy DNA in transcription, we only copy one side, though. We call that the sense strand, or the coding strand. The other strand is called the nonsense, or the non-coding strand. So when we talk about DNA becoming RNA, we are only copying one side of the DNA. There's 2 steps to make proteins in your body. The first is transcription, the second is translation. The first part happens inside the nucleus, the second happens in the cytoplasm. I'm going to go through today both of them and the notes, and then tomorrow we're going to draw the process together. So what is transcription? Essentially, transcription is DNA to messenger RNA. That's step 1. It's only happening in the nucleus, and DNA is coding for some proteins. So we need to get to that DNA. So what happens is we unzip the hydrogen bonds and we break them, much like you would see in replication. Except instead of helicase doing this job, we use ribosomal polymerase, or RNA polymerase to do the job. And what ends up happening is that polymerase adds free RNA nucleotides. Just like a polymerase, DNA polymerase adds DNA in replication, RNA polymerase adds RNA during transcription. So it adds it to the complementary base pairs of the DNA, the coding strand or the sense strand. Then what happens is mRNA codons are synthesized, so they're kind of all put together, and then they exit the nucleus. So it kind of looks a little something like this. You have your RNA polymerase. It's opening it up in the middle of the DNA. It's going to nicely open it up. You have your coding strand, your template strand, okay? And you're going to copy it. So here's my coding strand, A, T, G, A, T, C, so forth. You will see as it copies it, anytime there is a T somewhere, oh, sorry, here's the copying strand. Anytime there's an A, it'll replace it for a U. So anytime RNA is being made, if there is an A, you do not pair it with a T. RNA uses U's. So within translation, which is the second step, we are now going to make amino acids. So this happens in the cytoplasm. And what happens is the mRNA leaves the nucleus and enters to the ribosome, which is hanging out in the cytoplasm, and it's waiting there for tRNA and mRNA. As it's waiting, mRNA loads itself into the ribosome, and then tRNA is signaled, hey, start bringing those amino acids in. Now, the interesting thing about RNA is that it starts with an initiator codon, usually AUG or methyladenine. Methyladenine, if it's found in the middle of RNA, so if you see AUG in the middle of a long RNA strand, we call it by its regular name. But if you see it at the very beginning, we call that a start codon. Same with there's a bunch of triplets that end the sequence. There's UGG, UAG. They are called stop codons. And essentially their job is to make sure that the ribosome knows to stop manufacturing the proteins. So translation, mRNA to our polypeptides, is all determined by which amino acids are brought to the mRNA. Now, you have to use your data sheet for this. So that's why I said that data sheet is going to become extremely important. During this time, we are going to figure out what specific amino acids meet with the mRNA. So here's mRNA. This is called a codon, 3 letters together, GUC. When I go to my Bio30 data sheet and I look at my amino acid chart, I'm looking up GUC. I will notice GUC matches with valine, and that is my very first amino acid. I follow that process through the whole thing. So now I have UCA, then AAG. I am going and I'm finding the corresponding amino acids from the chart that you have. Now, tRNA is kind of a complicated idea because it's really just this random thing. But what it is it, like I said, it's like the messenger. It's like the invitation to the party. tRNA is the one that gathers the amino acids and then brings it to the mRNA. Now, how does it know what mRNA to go to? Well, tRNA we call anticodons. So like DNA has 2 complementary strands, RNA technically has complementary strands just in the form of mRNA and tRNA. So this is how it works. You can see that I have a non-coding strand and a coding strand. I made my coding strand AUG into mRNA, so AUG, GGC, GCU, and all forth and forth. Those amino acids match that. But I could have thrown in a step here that says, tell me tRNA. If you were to tell me tRNA, tRNA is the complementary strand to mRNA. So instead of AUG, I would have UAC, CCG, GCA. It's just the complementary strand. tRNA is just complementing the mRNA and attaching the amino acid in the process. There are only 20 amino acids in our bodies. Okay, so that means there's only so many anticodons. All right, so those complementary strands, there's only so many of them. Okay, the ribosome itself is just acting as a scaffold. It holds everything together and starts building that protein. So again, here's just another example of how messenger RNA has a complementary set in tRNA and the amino acids built. Now, it's very confusing because most students will look at the tRNA and go AAU and go to their chart and build AAU. Do not use tRNA to build any amino acids whatsoever. The only way you build amino acids is with your mRNA, UUA. UUA is what matches leucine. This guy here is just the invitation to the party. It's just carrying it over, okay? So whatever you do when you're using your sheet, do not look up the transfer RNA. In the end, you got to package it all together. So we know that primary structures are a bunch of amino acids put together. They're big, big chains. Then what eventually happens is we start packaging into secondary, tertiary, and quaternary structures. Most tertiary, quaternary structures are what you're going to see inside your body, okay? The Golgi apparatus, which you may remember again, way back from Science 10, is where we package our proteins and put it all together in a lovely little rounded out glob, essentially, I guess you could say. So those are the 2 processes of protein synthesis and the 2 processes of transcription and translation. Put this all in your notes, and hopefully tomorrow when we do the process as a picture, you will have a better idea of what it looks like as we go through. The last thing I want you to do is after you've taken your notes down, there's a little bit of practice after that says determine the amino acids produced by the following DNA coding strands. And then you have a couple coding strands. I want you to try those 2 questions for practice. I will come to them in the video tomorrow.